So this is Ethan Booth from our analysis team here in Bangkok, and is going to present some of our results from the vivax pipelines. Ethan.
Hello, everybody. I'm Ethan Booth. I'm a research scientist for the GenRe-Mekong project. So I'll be talking about the progress we're making on the Plasmodium vivax genetic surveillance in the project and the results we're getting so far. So to start, as Sarah Auburn mentioned, the cases of malaria have been reducing quite significantly in the GMS. Most of this reduction, however, comes from the reduction in Plasmodium falciparum cases, while vivax cases has remained quite level. And so this means that the proportion of vivax cases has increased in recent years, which is a challenge for our national malaria control programs because vivax is much more resilient to control activities than falciparum. And so control activities need to be more tailored to vivax elimination. And one of the key activities of elimination is surveillance. However, when surveillance is complemented with genetic data, it's much more powerful. Genetic surveillance can be used to address questions such as those pertaining to the epidemiology of drug resistance. It can be used to address the origins of outbreaks and characterizing outbreaks. And it can also aid in facilitating certification, for example, by determining imported cases versus local cases. However, because Plasmodium vivax, as of now, there are no validated genetic markers for drug resistance, the use cases for genetic surveillance focuses on outbreak investigation and determining imported cases. So to this end, the GenRe-Mekong project has started genotyping vivax samples beginning in March of this year. And since then, we've genotyped a total of 1,320 samples coming from Vietnam, Cambodia, and Laos. And this is all thanks to the microhaplotype panel from the Menzies group, which I'll get into in a little bit. And so the panel was designed, and Sarah Auburn explained this quite clearly, with genetic diversity in mind. So it's designed to capture the highest diversity of PV globally. So I want to explain first what a microhaplotype is, but also how it affects the GenRe-Mekong data outputs as well. So because a microhaplotype is a sequence of SNPs for a particular region of the genome, a single microhaplotype contains much more information than a single biallelic SNP. However, the challenge here is that we can't really anticipate how many alleles there are in the population compared to a biallelic SNP, which there are just two, the reference allele and the alternative allele. And so this is a problem because the GRC, the genetic report cards, only have only had 101 barcoding SNPs. And these are fixed across all the GRCs. However, to accommodate this new data type, we must either make variable the number of barcoding SNPs in the GRC. So with each new batch, new SNPs are discovered. And so these new SNPs are then incorporated into the GRC, making the list of SNPs grow. Or either we fix the number of targets, so 93 targets, and make variable the number of alleles for each target. So to address this, we've updated the genetic report cards. So in the past, the genetic report cards contain a fixed number of barcoding SNPs, but with the GRC 2.0, we have designed the GRC to make it descriptive. So there are now columns that reference other columns in the GRC, which makes it now more flexible. So we can add, remove, or modify columns if we need. So with the most recent PVGRC, we have 768 barcoding SNPs and a total of 4 putatively associated drug resistance alleles, drug resistant mutations. So let's get into the quality of the PVGRC. So as you can see, the quality of the PVGRC when it comes to missingness, which is the proportion of missing genotypes for a sample, is lower than the PFGRC. Actually, so most samples have less than 20% missingness. And of the samples that have more than 20% missingness, most are failed samples rather than samples with intermediate levels of missingness, unlike PF, where there's a much higher proportion of samples with an intermediate to high level of missingness. In addition, the heterozygosity of these samples, which is the proportion of heterozygous genotypes for all the barcoding SNPs per sample, is also low. It might seem higher, but that's only for lower levels of heterozygosity, between less than 10%. But when you look at heterozygosity levels above 10%, the PV actually has a lower proportion of samples in this category than PF. So to sum up, the PV sample quality is excellent for both for missingness and heterozygosity. Now, looking at the barcodes instead now, I've excluded missing samples with higher than 75% missingness for this analysis. And now we're asking the question, for a given barcode, how many samples have a missing or heterozygous genotype as a proportion of all samples? So the missingness of PV is actually an order of magnitude lower than PF. So remarkably low missingness. For heterozygosity, heterozygosity is comparable with PF. So the quality of the PVGRC is quite good. And we're getting excellent results from it as well. So looking at the distribution of samples, we have a roughly equal distribution of samples coming from 3 countries, so about 450 samples per country. These samples range from 2018 to 2023, and they come from a total of 21 provinces over 63 districts. So a good representation of samples in the region. Moving on to the drug resistance associated SNPs, we are monitoring for 4 mutations in total, 2 in PVMDR1, which is associated with chloroquine resistance, and 2 in PVDHPS, which is associated with sulfadoxin resistance. In PVMDR1, the 2 mutations we're looking at are Y976F and F1076L. So you can see here, actually the proportion of wild-type parasites, wild-type for both mutations, the YF haplotype, has actually increased in the region. And you can see this most notably in Vietnam, where in 2023, the majority of samples are wild-type. You can see this to a lesser extent in Cambodia and in Laos. Now, I've mapped the distribution of the Y976 mutation to see if the geographical distribution of the mutant. And you see that the change isn't actually evenly distributed. So it's patchy actually. So there's an area in southern Laos which shows a steep reduction in the level of the mutant. So in Savannakhet, for example, which fell from 92% of samples between 2018 to 2022 to only 18% of samples in 2023. Steep reductions in Champasak as well. And another region in southern Vietnam, which previously shown very high levels of the mutant, dropping to 0%, so almost all wild types. In Cambodia, the levels remain pretty stable at 50 and 40%. So for mutations in the DHPS gene, we are monitoring the A383G and A553G. However, for these sets of mutation, the wild-type parasites have dominated the region and continue to dominate the region until the most recent time point. Now, looking at the diversity of PV in the region, so here we're looking at the genetic distance, the pairwise genetic distance between every sample in the region and then between every sample in individual countries. The mean distance is 0.255, but when we break it down by country, we actually see heterogeneity. So in Cambodia and Vietnam, the diversity is similar, but in Laos, the diversity is lower than the two other countries. And this is excluding these samples here, which are clonal. Speaking of which, you can see these tails here, which show the pairwise distance between the highly related samples, much more so in Laos and Vietnam than Cambodia. Now, mapping the diversity in the region, I use the maximum haplotype frequency as a measure, which is a measure that indicates the frequency of the most common haplotype at a location. So, for example, 60% in Salavan means that 60% of the samples in this province share exactly the same haplotype. So it's a good indicator of clonal expansions. So looking at the map, we can highlight two areas of low genetic diversity. So there's this area in central Vietnam and some provinces in Laos and in southern Vietnam. And to confirm this, I used another measure of diversity, which is the mean SNP heterozygosity. With this measure, the value of the measure is actually positively correlated with diversity. So the higher the measure value, the higher the diversity. So it also confirms that these two regions have a lower diversity than the rest of the provinces. So there are a lot of clonal expansions going on in the region, but what is the geographical distribution of these expansions and clusters of related parasites? So to investigate this, I clustered the parasites at a 95% genetic identity using the genetic barcodes. And so almost half of the samples are unclustered, so they're more related, distantly related to each other, while half of all the clustered samples belong to smaller clusters, so clusters of less than 10 samples. These large clusters of more than 10, were seen in all three countries, which indicates several clonal expansions. And mapping the top 10 clusters at 95% identity, you can see that most clusters actually remained in a single province, while two clusters actually circulated across two countries, while two other clusters circulated between two provinces, in a single country. So the largest cluster, the cluster in light blue, circulated between Samanaket, Salavan, and Sekong in Laos, and Quang Tri in Vietnam. The fourth largest cluster in green circulated between Samanaket and Quang Tri. The two clusters that circulate between two provinces in Vietnam are cluster 5 and 6. And so even with a high genetic similarity, we still see clusters that circulate over quite large geographical distances. And looking now at the families of related parasites, so we loosen the genetic identity threshold to 80%, and we find results that are quite remarkable in that we see these clusters that sprawl across the region. For example, the first cluster, we can see between Bin Phuc and Nga An. I hope I'm pronouncing that right. But what this means is that a parasite from this cluster in Bin Phuc is more related to a parasite in Nga An of the same cluster 760 kilometers away than it is with another parasite in a different cluster in the same location collected in the same time, which is pretty remarkable. And you can see this for not just the largest cluster, but also the smaller clusters as well. So here, I'm showing you maps of the top 8 cluster, and you can see even with the smallest clusters, the 8th largest cluster here, 36 samples, it sprawls across Cambodia and southern Vietnam. So really interesting, and we're going to dig into this to see what's happening. So to conclude, we have developed and are developing tools to now allow analysis of microhaplotype data. The quality of the barcodes and of the samples are excellent and comparable, at least comparable to PPF. There's a fall in the Y976F mutation, while the wild-type DHPS dominates. There are pockets of low diversity in the regions, especially in central Vietnam and in southern Vietnam, which indicates clonal expansion. And when we map these expansions, we see that these expansions actually circulate between more than one country, especially between Quang Tri and Samanakhet, and that there are families of related parasites that are more related to each other far away than they are with other clusters in the same area. So that's all for my talk. I'd like to acknowledge the Bill and Melinda Gates Foundation, the Global Fund, and the Wellcome Trust for supporting us. Thank you.
Thank you, Ethan. Thank you, Ethan. So I think these analyses actually are a sort of data that's never really been seen before. The genetic relationship at such detailed level between provinces of the Eastern GMS. And we were quite surprised, and Ethan pointed it out, we were quite surprised that of the level of similarity that you can get in parasites that are across the border. As Sarah said, with the microhaplotypes, it's sort of fairly easy to distinguish between individuals, and having 95% similarity is a huge, essentially you're going into the area of genotyping errors, right? Otherwise they are identical. So to see that between adjacent provinces of different countries, and also at quite significant distance, that you have essentially identity of parasites, gives a new dimension to how we look at this vivax population. And I wanted to just quickly add, Dr. Keo, earlier on, Sarah talked about the Nakai outbreak, and actually we were able to then put these samples into these data and compare to the current samples both in Laos and Vietnam. And so this sort of big tree, this is 1,500 samples from the 3 countries. And you can see over here that these yellow ones here, these are the Nakai samples. You can see they're all clustered together, so they're almost identical to each other. And the interesting thing is they all part of a branch where you find both samples from Laos, from Attapeu, but also from Vietnam, from Quang Binh province, which is next door to Cam Ranh province. So these are definitely local parasites. So that's probably a very good thing. I don't know. But the interesting thing is they're not isolated. They're part of a family that has been living there for a while, basically. Yeah. So, but actually, the reason I'm showing you this is now we have quite a high resolution power to look at the origin of these outbreaks and compare them to other samples that you've all contributed from other provinces and so on. So this is now quite a powerful way of analyzing the data. Sorry to have barged in like this. Can I take any questions for Ethan? Yeah? Yes. Oh.
Okay.
Yeah.
Thank you for very interesting about the Plasmodium virus diversity. And I have some question about for you. For you about first thing I want to ask about the haplotype because for haplotype. So what thing you looking for to find haplotype? I mean because you also talk about the pair coding of Plasmodium virus. So how to differentiate about the SNB SNP and also haplotype, and in your presentation you talk about 10 you find 10 cluster in your on the sample from GMS, right? So and I because you talk so much data, so I want to know how to differentiate cluster and also haplotype because what involve in cluster and haplotype, something like that. Yeah, that's
So we don't really use haplotype data yet. I haven't analyzed the haplotype data as it is. So what we've done is we've broken the haplotypes into SNPs, and then we generate the GRCs that way. But we do have a data set of PV with the microhaplotypes, and like from a preliminary look, they seem to match up with the results from PV, the SNP. But with clustering, we use a algorithm called the Louvain method to to cluster samples with a similar barcode genotype. So, for example, at 95% means that 95% of all the SNPs, all the alleles in the SNP, if they're similar to another sample, they will be clustered together. So at 80%, for example, two samples can be clustered together if they share 4 out of 5 SNPs, for example.
Actually, this is the same software algorithms that are used, for instance, to group together people who go on Facebook and they know each other. You know, it's the same. It's actually same concept. So you look for networks of closely related samples, and the algorithm finds the clusters.
Well, I've just shown you the 10 biggest clusters, but we actually found a lot more.
Yeah, so I just want to know about how many haplotypes you find in the sample from cannot find how many.
How many total haplotypes?
Haplotype.
Oh gosh, so many, hundreds, I would say.
Yes,
Yeah.
yes.
Yeah.
And because you also talk about the less diverse from Plasmodium virus, the less diversity than Plasmodium falciparum, right?
More diversity.
More or less.
More.
Yeah.
Well, actually, we can't.
Okay.
From previous literature, we know that vivax is more diverse, but using the SNPs, we can't actually compare them using these SNPs because they're not comparable. Yeah.
Sorry, one more thing to say about there's different ways of looking at diversity. But one thing that we noticed is that our vivax samples have less heterozygosity. They have less mixed infections than the falciparum ones, which I think goes against what has been published before. You know, I always heard, there's a lot of mixed infection in vivax, but we see more in falciparum than in vivax. Yeah. Quite a bit more, right?
Yeah.
Yeah. Yes.
Thank you for nice presentation. I just wanted to ask you, the data you took for analyzing, when is that?
When?
Yeah, because in which
So between 2018 and 2023.
because you saw the similar between two provinces in Vietnam, Nghệ An and Bình Phước.
Yeah.
So in the past maybe reasonable because maybe some people they move from Nghệ An to Bình Phước for seasonal working.
Yeah.
But recently it's completely different because most of the malaria cases from Nghệ An is imported from Africa.
Oh. Okay.
So you saw
Yeah.
that the similar.
Yeah.
Well,
I
I think that is something that we haven't done, so to layer the analysis by year. I think
Yeah.
we need to do that.
Yeah.
You're quite right.
yeah.
Yeah. Thank you very much, Tang. That's a very good point, actually.